this stuff, so I'll go over it pretty quickly. But transcription, when DNA is unzipped and it's copied to form an mRNA strand with complementary base pairs. Translation, mRNA strand travels to the ribosome, and tRNA brings amino acids to make proteins. So you guys have learned all that, right? You guys are good with that? Okay. This is just visual. If you guys need any more refreshing. That big cloud right there is the ribosome, and then those little dots. It doesn't show the tRNA, but the tRNA is a molecule that will bring those dots over to the matching mRNA strands. So you guys know all that. DNA is found, a lot of people don't know this actually, DNA is found, like, instead of being in little strands, it's coiled up really tightly in these big shapes called chromosomes, and those are found in the nucleus of the cell. And histones, I know you guys have talked a little bit about these, but histones are proteins that the DNA is coiled around, and lots of histones form chromosomes. And then this picture sort of it gives you a good visual of epigenetics. So right here you can see the chromosomes are sort of loose, and then the DNA is not coiled around it, but this is how it actually looks. So the DNA is coiled pretty tightly, and then there's those big spaces, but normally those are coiled up too, to form supercoils, which eventually form a chromosome. So you guys can imagine, how can an enzyme get in there to translate or transcribe the mRNA that it needs to make proteins and other things like that. So it does this through a process called epigenetics, and epigenetics literally translates to above the genome, which means that it can, like, there are ways that outside factors can affect your genome without you altering the sequence. So histone activity is controlled by methyl groups, which is a group of organic compounds that you get when you eat certain foods. So when methyl groups enter your body, they can attach to histones, which can cause them to coil up more, which would mean that you have more coiled DNA, so that enzymes can't get in there and transcribe. So, I mean, like, consequently, this means that these genes won't be expressed, so certain proteins may not be expressed, or certain proteins may be expressed with a lack or a presence of methyl groups. So a lot of examples of how epigenetics can really affect you is, like, for example, I know Ms. Wilson mentioned that there's a twin in here.
A couple of times.
The reason that you and your twin might have, like, different health factors, or like other, like, different behavioral factors, it could be an example of epigenetics, where what you guys are eating may affect certain genes, like turn off certain genes, so gene expression can affect, like, what's, like, certain proteins are being made and stuff like that. So in this experiment, one twin had diabetes, one identical twin had diabetes, and one identical twin did not. So scientists were experimenting with tagging the methyl groups on each twin's DNA, and they found that the methylation was really different, very varied between the twins. So they haven't concluded, like, they haven't strongly concluded that, like, certain foods or certain behaviors are causing these certain expressions within the twins, but they have linked it back to epigenetics. Alzheimer's research also suggests that the cause of the cure, or of the disease, might potentially be related to certain methylation in DNA, but I mean, these aren't, like, conclusive results. They're just sort of recent studies. And then tumor cell research also suggests that the productivity of tumor-fighting cancer cells may be compromised by lack of expression due to epigenetics. So certain proteins that could be produced to fight cancer cells, fight tumor growth, are being silenced by the lack of this methyl groups. So yeah, there's not a lot of research done about which foods could give you more methyl groups or not, but could do if you're interested in that. Okay, that's it.
Do you all have any questions? Can I ask you to help clarify one thing for them? So you mentioned the methyl groups. How does that relate to the coiling?
Yeah, the methyl groups, they'll attach to the histones, and the presence of more methyl groups means that the histones will coil up more tightly. So less methyl groups will mean that the histones are farther apart, and the DNA strand is more exposed. So enzyme could come and transcribe this strand more easily than if there were more methyl groups and the strands were more tightly coiled, and the gene would consequently be sequenced because the enzyme couldn't come transcribe.
Good. And so you get more or less methyl groups based on what you're eating or other factors like stress and what you're saying. Okay, great. All right. Thank you very much for coming to visit.